Here, we will demonstrate how to expand human airway epithelial cells and initiate cultures at the air-liquid interface, or ALI. The ALI workflow involves 3 phases: expansion culture in a flask, submerged expansion in inserts, and ALI culture in inserts. In this video, we will focus on phase 1 and 2, the 2 expansion steps of the workflow. Phase 1. Expansion culture in a flask. To begin, thaw PneumaCult EX plus 50X supplement at room temperature. If a precipitate is observed after thawing, mix the contents by inverting the vial a couple of times. The precipitate will return to solution and will not affect the culture performance. Use the supplement immediately, or aliquot and store at -20°C. Prepare 500 milliliters of complete PneumaCult EX Plus medium. Mix the components thoroughly. The complete medium can be stored at 2 to 8°C for up to 4 weeks. To initiate culture, obtain frozen primary airway cells such as bronchial, tracheal, nasal, or small airway epithelial cells. These can be from commercial sources or donor lung tissue. If using freshly isolated cells, additional preparations may be required. For plating, only use tissue culture-treated cultureware to ensure that cells adhere optimally to the surface. Prepare a T25 square centimeter flask with 5 milliliters of complete PneumaCult EX Plus medium and pre-warm at 37°C. Thaw the frozen cells by following the protocol recommended by your vendor. Use the viability and plating efficiency data from the Certificate of Analysis to calculate the volume required to plate 300,000 cells, or 12,000 cells per square centimeter. Transfer the cells directly into the pre-warmed medium and mix them well. Gently rock the flask back and forth to ensure that the cells are evenly distributed across the culture surface. Incubate the cells at 37°C. And perform a medium change roughly 18 hours after initial plating. Perform medium changes every 2 days until the cells are ready to be passaged. On weekends, change the medium on Friday afternoons and Monday mornings. The expanded cultures display a colony-like morphology. The cells are small and become more tightly packed at the center of the colony over the course of expansion. At 50 to 60% confluence, the cells are ready to be passaged and can either be differentiated, further expanded, or cryopreserved. Typically, most cell populations take 3 to 5 days to reach 50 to 60% confluency. When culturing in PneumaCult EX Plus, confluence should not exceed 70%, as this can negatively affect the differentiation potential of the cells. On the day of passaging, remove the old medium from the flask and wash the cells once with 5 milliliters of room temperature PBS. Add 2.5 milliliters of room temperature ACF enzymatic dissociation solution to cover the surface of the cells. Next, incubate the cells at 37°C for 7 to 8 minutes. Observe the culture under the microscope to confirm that the cells are detached. The cells can be dislodged from the culture surface by firmly flicking the flask as needed. Add 2.5 milliliters of room temperature ACF enzyme inhibition solution. Wash over the culture surface using the entire cell suspension to capture any leftover cells. Transfer the cells to a polystyrene centrifuge tube. Centrifuge at 350 G or 1200 RPM for 5 minutes. Discard the supernatant and resuspend the cell pellet in 1 to 2 milliliters of pre-warmed, complete PneumaCult EX Plus medium. Perform a viable cell count with trypan blue and a hemocytometer using an inverted microscope at 10 times magnification. Next, determine the cell concentration and calculate the volume needed to plate at the desired density. The dissociated cells can be seeded into a flask for further expansion, into a transwell insert for differentiation, or cryopreserved for future experiments. If expanding further into a T25 flask, seed at a density of 125,000 cells in 5 milliliters of pre-warmed PneumaCult EX Plus medium. Note that primary airway epithelial cells will gradually lose differentiation potential with extended passaging. If cryopreservation is desired, CryoStor CS10 can be used during the expansion phase only. We recommend freezing down airway epithelial cells at a concentration of 1 million cells per milliliter. Cells cultured in PneumaCult EX+ maintain widespread expression of basal cell markers as visualized by immunocytochemistry. Phase 2. Submerged expansion in inserts. To initiate the submerged culture, start by preparing the culture plate with a transwell insert. ALI cultures generated using PET membrane co-star transwell inserts with a 0.4 micrometer pore size can achieve a highly successful differentiated morphology. If desired, individual transwell inserts can be manually transferred into a new culture plate using a pair of sterile forceps or a sterile pipette tip. The original plate containing the inserts can be parafilmed and stored for later use. In a 24-well plate format, add 200 microliters of pre-warmed PneumaCult EX+ medium into the transwell apical compartment. Then seed 100,000 cells per square centimeter, or 33,000 cells, into the apical compartment. Next, add 500 microliters of pre-warmed PneumaCult EX+ medium to the basal compartment. Incubate the cultures at 37 degrees Celsius. Then, perform full medium changes on both the basal and apical compartments every 2 days until confluence is reached. This typically takes 2 to 4 days. The confluence on day 1 will depend on the seeding density and the number of cells that adhere to the insert. When the cells reach 100% confluence, the cultures are ready to be airlifted and cultured using PneumaCult ALI medium to achieve mucociliary differentiation. For the next step in the ALI workflow, proceed to Video 2, Differentiation of Human Airway Epithelial Cells. For more information about our PneumaCult products, visit pneumacult.com.